Okay, so now we look at this fractionation. So from the name fractionation, basically we want to separate the cells, organelles, or cellular components. Okay, so what's the function of fractionation? Fractionation actually is a process we use to separate the cellular component. Okay, and at the same time we want to preserve their functions. Okay, we don't want to break entire thing, but we want to isolate each organelle. So typically we get the tissues and then we homogenize. Homogenize means that break the cell. Okay, we break the cell wall. For the plant cell, we break the animal cell membrane, and we place them in an osmotic buffer solution. Now, what does mean osmotic buffer solution? Now, try to imagine, guys, if this is my beaker. So we have the cell suspension inside. So we want to actually, or tissues, cells or tissues. We want to do here, actually, we want to homogenize it. Homogenize basically, we want to lyse the cell to break the cell. But the problem here is we do know that if we lyse it inside the distilled water, if this is a cell, and this is a distilled water, what will happen here is distilled water we're going to break the cells and break the organelles. So therefore, we won't be able to study each organelle. At the end, we get all the lyse one, breakdown one. Okay, so useless. Okay, so once we homogenize it, it will form a thick homogenate. So liquid suspensions of the crushed tissue sample. So how we can actually homogenize it? So we can use this few method. We can grind it. We can use a pressure change, or we use the sonicator or ultrasonic vibration. We vibrate until the cell membrane break. Okay, then release all the organelles. So we need to control the ultrasonic vibration. We need to control a certain pressure so that we only break the cell membrane, break the cell wall, releasing these organelles. And once you form the homogenates already, okay, then we need to place these homogenates on ice to keep cold so that reduce enzymatic damage. Because when the cells lyse already, immediately going to trigger the enzymatic reaction to destroy the organelle. So our idea here is to preserve the organelle so that I can study the organelle. So if you lyse everything, you have the enzymatic reactions to break down all the organelle, I won't be able to study the functions of each organelles. Are you clear? Of each component. So first, what we do, we place the tissues in the homogenizer. For how we can do it, we can grind it, we can do the pressure change, we can actually use ultrasonicator. What to do? We actually lyse the cells, and then we lyse it inside osmotic buffer solution so that each individual organelles can be preserved and it won't be lysed. And then it must be on ice or keep cold to reduce the enzymatic damage. Be careful, guys, we cannot stop, but we can reduce the enzymatic damage. Okay. And then the homogenate will be subjected to what we call the differential centrifugation. Now, what's mean centrifugation? Now, very simple thing. Okay, very simple. Look at this. If I have a tube of homogenate, so these two are homogenates. Okay, what will happen here is we want different organelles sink down. Okay, if you sink down, okay, I get the first organelle, and then sink down, get the second organelle, sink down to get the third organelle. So very simple thing here is you try to imagine that you get a tube or a bottle. Okay, then what you do, you place the sand at different, okay, sand at different, okay, you put a stone, you put the sand, you put the water in, and then you shake it, and then you leave it. When you shake it and leave it, stone will sink down first, correct? Stone will sink down first, and then followed by the sand, correct? Okay, because stone larger, higher density, correct? But stone can sink down is because of the gravity. Gravity has the sufficient force to pull the stone to sink down, sand to sink down. But the dust may still disperse, correct? So it depends on the gravity. But the organelle is too small. The gravity may not have the effect. Yes, we have the effect, but if you want to wait, you wait for too long. Are you clear? So what we do here is we use a machine called centrifuge machine. So these centrifuge machines or ultra centrifuge machines, when you spin something, it will create what we call the G force. Okay, we're going to exert the G force or gravitational force onto this. Means that when you spin it, you increase the G force. So when you have this G force or gravitational force, you will be able to pull the organelle down to the bottom of the tube, and then from the tube we can collect and do the study. Okay, so based on what? So since different fragments of the cells have different size and the density, so each fragment will settle into the pellet with a different force. So how we change the force depends on how fast we spin. Are you clear? How fast we spin. So the faster we spin, then the organelle will sink down faster. Are you clear? So the different sedimentation rates of various cellular components make it possible to separate them partially by centrifugation. After centrifugation at each speed, for an appropriate time, the supernatant is poured off and centrifuged again at the higher speed. So each pellet fractions can be resuspended and further separated by equilibrium density gradient is another method that we need to continue to separate them. Okay. So now how we do this? Let me show you. In this diagram, what we're going to do here is we filter the homogenate to remove any clump or unbroken cells. Then we get this. So this one we call as a homogenate. So first we spin. Can you see that? We spin it. When we spin it, we create a, what we call this, the gravitational force. How high the gravitational force? It's about 600 times of the gravitational force. Are you clear? You try to imagine what we experience here is one time. So when we spin, we increase it to 600 times the gravitational force for 10 minutes. After 10 minutes, we will be able to separate out the first pellet. So this is we call first pellet. Are you clear? And we have the supernatant. Supernatant means that there are fluids. And the remaining organelle will be stay inside here. Nucleus is the largest one. So that's why nuclear heavier. So with 600 times, 600 times, then you can see that we already able to get the nucleus out. So this is pellet 1. Okay, pellet 1. So then we take this supernatant, pour into a new tube. Are you clear? You pour this liquid into a new tube. Now, we cannot use back 600 because just now we use 600, we cannot get the mitochondria out. We cannot get the chloroplast out, right? We cannot get the ribosome out. So it means that I have to increase the speed. Increase the speed so that I can increase the gravitational force. So we pour out these liquids and then we spin it again, but we increase the speed to 15,000, okay? 15,000 times the gravitational force. Are you clear? So at this speed and 5 minutes, after 5 minutes, we will be able to get mitochondria out, chloroplast, as well as lysosome. Are you clear? Okay, it depends. Okay, at this pellet, we call pellet 2. But most of the times, you have to be careful because in a plant cell, they don't have lysosome. So they only have mitochondria and chloroplast. Pellet 2, very, very important, guys. Pellet 2, you have to be careful whether the sample is the plant sample or animal sample. If let's say it's a plant sample, then you only get the chloroplast and mitochondria. Are you clear? Okay. But if let's say it's an animal sample, you may get mitochondria as well, and you will get a mitochondria and the lysosome without chloroplast. So for pellet 2, always be careful, okay, when you write. So check whether plant cells or animal cells. Are you clear? Okay. So now next. Okay. So with this speed, 15,000, we only can get the mitochondria or chloroplast or lysosome out. Peroxisome is not in our syllabus. Peroxisome have a similar function as the lysosome. Okay, so we get the supernatant. So this supernatant, we pour it. Okay, supernatant, we pour it out. So we get the pellet. Then we increase the speed now. What increase speed? We increase to 100,000. Spin it for 1 hour. When you spin it for 1 hour, you will get the plasma membrane, but it's not the intact plasma membrane because we lyse it already. Are you clear? We lyse it already. So therefore, you can see that plasma membrane separated, and you can get what we call the microsomal fraction. What's microsomal? Microsomal is those organelles that made by the membrane. For example, endoplasmic reticulum, Golgi apparatus, those will be able to be separate out and form the pellet. Now, one of the questions here is lysosome. Lysosome can appear also in pellet 3. Are you clear? This is pellet 1, second pellet, and third pellet. Lysosome, not necessary in the second. It can also, because it depends on the fraction. Are you clear? It becomes what you say that when you spin it, so larger one may be a bit larger lysosome will be here, but smaller one will be in this. So these two. But generally, we put it as the fraction 2, generally. But sometimes some question may ask you pellet 3. Pellet 3 actually can get lysosome. I said lysosome a bit confusing always, okay? Be careful. Generally, number 2. Are you clear? But if you have the choice, let's say, for example, question asks you, which of the following organelle can be found in pellet 2? A, mitochondria. B, nuclei. C, lysosome. D, ribosome. Then answer will be mitochondria, because mitochondria only can get in the fraction 2. Lysosome can be 2 or 3. So if you choose between mitochondria and lysosome, go for mitochondria. Get what I mean or not? Okay, for example, I say that question asked, okay, which of the following organelles can be, present in pellet 2? A, mitochondria. B, lysosome. C, nuclei. D, ribosome. So answer will be mitochondria. Okay, be careful, not lysosome. Okay, because mitochondria confirmed will be in pellet 2, but lysosome can be 2 or 3, depends on their size. Okay, so what we can get it from the pellet 3, endoplasmic reticulum or Golgi apparatus. Yeah, membrane-bound organelles, we can get it here. Okay, then after we have, we can collect it, then this supernatant we pour into a tube. Okay, the new tubes, and then we spin it now with 300,000 gravitational force, the speed. Are you clear? Then about 2 hours, you will be able to get the ribosome or ribosomal subunit, either small or large subunit. Are you clear? So means that ribosome is the lightest organelle. So when you get it out already, the remaining one is just a solution without any organelles. So without any organelles is termed as the cytosol, not cytoplasm. Cytoplasm is equal to, okay, right now, cytoplasm equal to the cytosol plus organelle. So means that we already remove all the organelle out already, right? So the remaining ones is the cytosol. Are you clear? Okay. So we look at the question here. So generally, the cellular homogenate first filter a centrifuge at relatively low speed to remove the unbroken cells. Then centrifugation of the homogenate at a slightly faster speed or higher speed for longer durations. Then the largest organelle, usually 5 to 10 micrometer diameter, will be pellet out. So this one is the first pellets will be nucleus. Then we increase, okay, the speed that we use is 600 G, okay, for 10 minutes. So this roughly equivalents to 500 revolutions per minute. So what's this mean? Because you spin it, right? So how fast you spin? You spin 500 times per, okay, per minute. You spin 500 times per minute, then you'll be able to get this. So your hand will damage. Okay, so the undeposited material, for example, with in this case supernatant, is next centrifuge at higher speed. You cannot use back the same speed now because the same speed already cannot precipitate them out. So why you use back the same speed? You have to increase the speed. Okay, with a time 5 minutes, then you get the mitochondria, chloroplast, or lysosome. Okay, but majority of mitochondria, chloroplast. Subsequently, we increase the speed and the times. Then we have the plasma membrane and fragments of the endoplasmic reticulum. And then a force to increase in one to increase to 100,000, you require 50,000 RPM. 50,000 basically means that you have to spin 50,000 times in 1 minute. Okay, so in this ultra centrifuge, now for you to use this ultra centrifuge is very, very important. We need to pump out the air when you rotate it because the speed is so high. it will generate the heat energy due to friction between the air and the spinning rotor, the machine that spin your tube. Okay, so the recovery of the ribosomal subunit require additional centrifugation at higher speeds. Then we will get the cytosol. So do you need to remember the speed? The answer is no. What you need to know only what are the pellets and what are the supernatant contents after each pellet. Are you clear? Okay, so means that if I use the homogenate, I spin it at 600 G-force in 10 minutes, what you're going to get here, the pellet. So you will get nuclei. Okay, so the supernatant contents, you have the non-nuclei organelles and also cytosol. So we continue to spin it at a low speed. Okay, so supernatant from the previous row. Okay, so supernatants, then spin the 5 minutes. What we're going to get here, we're going to get mitochondria, chloroplast, or lysosome. We just need to know the products of the pellets and also the supernatant. So supernatant will get the microsomal, okay, microsome or microsomal components. Okay, so cytosol and also the microsomal component. And we continue to spin. Then we continue to spin. Then we're going to get the plasma membrane and fragments of the endoplasmic reticulum. And the supernatant will have the ribosomal subunit or ribosome. And also the cytosol. So if we continue to spin it again, then what we're going to have here is you're going to get the ribosomal subunit or ribosome, and supernatant now contains cytosol only. Okay, supernatant basically means that the liquid form. Pellet is more to solid already, solid. Then the remaining. So for example, you can make a Milo. Cannot. You let the Milo sink down. So this one is called pellet. Okay, in centrifugations lah, but Milo we don't call pellet, but generally you can imagine. Okay, and then this one is the solutions, right? So solutions here is called as a supernatant. Okay, so this supernatant will contain something else that cannot sink down. Are you clear? Because the gravitational force is not huge enough to pull them to the lower part of the tube. Okay, do you get the ideas now, guys? Okay, so what you need to know, not the process. Process is for information only, because I cannot tell you what like this without us telling you guys what is the process. So this process is the differential centrifugation. So if you use the differential centrifugation, what will happen here is you will increase the speed and separate out the organelles. So through this technique, you will collect the organelles in the pellet. So you need to know each pellet. So this is pellet 1, pellet number 2, pellet number 3, and pellet number 4. So supernatant, the most important one, this one, because students always put their cytoplasm. No, the supernatant here contains the cytosol. Okay, means just a solution, nothing already. Okay, so if a question asks what is inside the cytosol, yes, we have glucose, nutrients, ions, waste material. Yes, inside cytosol, but they are not organelles. Are you clear? Okay. But the question here is, we still cannot, by using this technique, we still cannot differentiate mitochondria and chloroplast. Let's say plant cell, mitochondria and chloroplast. Right. So how we can differentiate them? Now we have to look at these methods, followed by what we call the equilibrium density gradient centrifugation. Okay. So question won't ask you the name. Definitely question won't ask you the name because not in our syllabus, but question will use this name. Then you have to know what is this mean. Equilibrium density gradient centrifugation is something like what we have done previously in physics, where we actually create a different density. Okay, so that a particular thing sink to their own density, they will stop to sink down. Okay, example. Okay, I put the mercury inside here. Okay, mercury roughly, not mistaken, is 13. Okay, the density. Okay, then I put in here, let's say water, and then a layer of the oil. So each of them will have their own density, correct? Okay, each of them will have their own density. So if I drop something, let's say for example, I drop something, okay, object A, and the object A now floats here. So we know that the density of A is really lower than the oil. So that's why float. Okay, so if let's say I have object B sink down and then here, then we do know that object B actually, the density is higher than the oil, but less than the water. Correct? Okay, so if I have object C, then it will sink down here, let's say for example. Then we know that this C, the density is higher than the water, but lower than the mercury. Are you clear? Okay, so if I have the mixture, let's say for example, now, let me clear up this one first. So if now I have, so if I have a mixture now, a beaker of a mixture A, B, and C, initially I cannot differentiate them, right? But if I pour it inside here, then I can see that A now stay here, B stay here, C will stay here. Correct? Why? Because now we can differentiate them based on their density. So just now we know that, for example, lysosome, mitochondria, okay, in this case, go peroxisome because this is an animal cells, okay, the rat liver. So that's why you don't have chloroplast. Can you see that? Are you clear? So how we separate between the mitochondria and lysosome? Because they are at the same pellets, right? So we create the gradient. Okay, we increasing the density, okay, by using a sucrose. How we create it? Different concentration. So for example, we have 1.25 gram per cm cube, 1.22, 1.19, 1.15, and so on. Then we have the organelle fraction, this one from the pellet 2. From pellet 2, now you can see that at the pellet 2, because the rat liver, so we know that it contains lysosome and mitochondria, pellet 2, right? So what we're going to do, we spin it again. So when you spin it, the spinning speed will drag them down, but they will be stopped at their particular density. So mitochondria density definitely will be higher than the lysosome because more membranes in this case. Are you clear? Mitochondria density will be the same as the, I said will be higher than lysosome. So that's why it's sink down to their own density. So at 1.18 gram per cm cube, lysosome will be higher. So from here, we will be able to differentiate between these two organelles. Are you clear? But if you use the first one, differential speed, you won't be able to separate them. But now continue with the equilibrium density gradient centrifugation, then you will get the mitochondria and lysosome to be separated. Okay, now my question here is, how about mitochondria versus chloroplast? In terms of the size, both of them almost the same. In terms of the size, both of them almost the same. But which one have higher density? Right now, your answer. Which one? Have a guess. Chloroplast or mitochondria? And why? If you can answer me, it's better. Why chloroplast? Some of you give me chloroplast. Why mitochondria? The rest of you, any answer right now before I disclose the answer? I only want 1 to 2 answer here. Some give mitochondria. How about the rest? Type out your answer in the chat box now. Okay, some say mitochondria, some say chloroplast. Doesn't matter. Again, how about the rest? No answer. Don't know. Which one have the higher density? Chloroplast? Not so sure. So that's why I put the question mark there. Okay, mitochondria. Okay, got fluid inside. Okay, what else? Yeah, you can direct message me. So no one, chloroplast because got starch. Okay, good. I want you guys to think. Now think about it. We study about chloroplast, we study mitochondria. So which one? Because they are same pellet 2. Both of them come from pellet 2. Mitochondria, chloroplast, any reason? 50% larger, heavier. Larger, that's not necessarily heavier, okay, but it's good, I guess here. What else? Okay, so the answer, okay, the answer is the density of the chloroplast will be greater than mitochondria, okay, because they have more membrane. They're heavier actually, in fact, okay. If you look at mitochondria, they have the outer membrane and inner membrane. Clear enough? Okay, but chloroplast, outer membrane, inner membrane, and also thylakoid membrane. So means that in terms of the structure, they are heavier, even though in terms of the volume and size, they are almost similar. So density, definitely chloroplast will be larger, okay, or higher in terms of density compared to the mitochondria. But in terms of the size, the length, they are almost the same for chloroplast and mitochondria, but mitochondria slightly heavier. Okay, so if you use the plant cells, then no lysosome lah. Okay, then you can see that chloroplast will be precipitated out lower compared to the mitochondria. Are you clear? So maybe somewhere here for chloroplast. Okay, if you use the plant cells. Okay, any more questions so far? So I stop here. So again, guys, since I have to record it again in this part, so just to assure you guys, chloroplast here, okay, they have higher density compared to the mitochondria. The reason is they have extra membranes structure here, membranous structure known as the thylakoids that form the grana. Okay, so in terms of size of length, they are almost the same. We're going to learn this one next subtopic, where we're going to learn the prokaryotic cells. Okay, they do have the same, in this case, they do have the same type of ribosome, 70S ribosome. They do have the circular DNA and they do have the enzyme. So the one that make them different here, because the chloroplast have, right, the chloroplast, they have extra membranous sac here known as the thylakoid. Okay, so stop the recording.